It is my great pleasure to introduce Kyle Mangano, who is a native Rhode Islander who received his bachelor's degree from McGill University. He leveraged his proximity to Cambridge, Massachusetts, to work at two small biotechnology companies developing stapled peptides and nanoparticles. He then obtained his PhD from the University of Illinois at Chicago in pharmaceutical sciences. His graduate studies focused on elucidating the mechanism of action of protein synthesis inhibitors using next-gen sequencing, and Kyle joined Amgen's induced proximity platform as a postdoc in 2022, where he has been using chemical biology and genomic engineering to study targeted protein degradation. So we're very excited to hear about your work.
All right, thank you. And here, let me share the screen here. Okay. Okay, can everyone see the screen appropriately?
It's great.
Okay, yeah. Well, thank you for the introduction. Thank you for the invitation. I'm pretty excited to tell you about my research today. As a postdoc in the Emerging Therapeutics Modalities group of Amgen, I've been working on a proof of concept for VIPER-TACs, which leverage viral E3 ligases for disease-specific targeted protein degradation. I'd like to start by kind of highlighting a challenge in early phase drug discovery, which is the restriction of target space. So, when considering a protein to treat a specific disease, let's say you have a great a GWAS analysis that really nails down one or two specific proteins that look good. You may run into some major limitations here. I think there's two that come to my mind. So the first is that many disease targets are not druggable or traditionally considered druggable. So there might not be enzymatic activity of the protein, or there might be extensive interactions with DNA or large protein-protein interfaces that are thought to be difficult to disrupt. Second challenge would be on-mechanism toxicity. So in this case, I like to think of kind of two examples. There's lots of proteins that have dual functions in the cells. And so, if you want to inhibit beta-catenin's Wnt signaling for certain cancer indications, you're likely to also disrupt its role in cell-to-cell adhesion, which will cause, distinct toxicities. And then there's also the essential proteins. If you want to kill cells, those are certainly a great targets to consider. There's about 1,000 of them, but you're going to have clear selectivity issues and potent toxicity with that as well. So luckily, there are some solutions to expand the target space. I think this webinar series is very familiar with targeted protein degradation. Of course, how it enables the drugging of the undruggable, by degrading instead of inhibiting proteins. So I think a classic example might be to this as an avenue of drugging transcription factors. But I'm not going to go into additional rationale here, but I'd just like to highlight that on-mechanism toxicity is still a concern with TPD because currently the majority of the E3 ligases that are used in TPD approaches are broadly expressed across cell types and tissue types. And so target degradation in healthy cells can lead to toxicity. And additionally, off-target degradation, so degradation of homologs or degradation of different proteins due to lack of specificity of the target ligand can still cause toxicity. So really to get around that, your therapeutics need disease-specific action. And so there's a couple ways to go about this. If you're lucky, you might have a disease causing mutant, which is druggable. So you could think of like the KRAS G12X mutants, where you can design therapeutics to specifically bind the mutant forms of KRAS while sparing the wild types. And then there's also targeted delivery. So that's like physically getting your therapeutic only to the diseased cells. And but there's one other option that kind of combines both of these solutions to treat both of these or to overcome both of these challenges. And that would be if you have more selectivity of your E3 ligase expression. So I think an area of research ongoing in the field is to look at tissue specificity of E3 ligases, and then also if there's any upregulation of specific E3 ligases in certain disease types. But really, I really wanted to ask, are there any truly disease-specific E3 ligases? And this is where viruses come into play. Because while humans have been investigating TBD for about 20 years, viruses have been doing this for like potentially billions of years. And I just really want to highlight that viruses have evolved to encode for proteins with E3 ligase activity. And these proteins are expressed upon infection, and they really act as biological PROTACs. So they bind and hijack the host E3 ligases and host ubiquitin proteasome machinery, and they recruit neosubstrates for ubiquitination and degradation. And they really do this just to enhance their replication, like they're the only thing they're kind of evolved to do. Going through the literature, I made this table of putative viral E3 ligases. Some things I really want to highlight are that viruses across all different groups encode. There's like representative viruses from all groups that have E3 ligases. They hijack different host E3 ligase machinery, in diverse ways. However, I will note that the culin ring ligases are overrepresented here, especially cul4, which is a potentially interest. And then for the neosubstrates, there's also a wide variety of those as well. However, I think a common theme is that they tend to be innate immune factors and cell cycle regulators. So this makes sense because if you're a virus, you want to hide from your host immune system. And in terms of cell cycle regulation, you might want to, like, if you're a DNA virus, you want to freeze the cells in a cell cycle where they're constantly replicating DNA. But this can have the byproduct or side effect of some viruses being oncogenic, which we're going to touch on later as well. So after coming up with this list, we kind of had the question, are any of these viral E3 ligases capable of targeted protein degradation by humans? So can we chemically hijack the hijackers to selectively treat disease cells? Here's a cartoon schematic of how that kind of therapeutic would look. What's absolutely critical here is the green circle ligand, because if you have a selective ligand, to the viral E3 ligase, you won't have any degradation in healthy cells, be on target degradation or in theory off target degradation as well. And so this alone is going to improve the therapeutic window of any TBD modality, but it critically also opens the target space to even any protein you'd want, including pan-essentials. And that's where we get the name VIPER, which stands for viral E3 pan-essential removing targeting chimera. Now that's kind of great in theory, but there's one major limitation, which is the almost complete lack of green circle ligands in the literature. And I think we can all appreciate that finding ligands to human E3 ligases is pretty challenging. And I think it's even more so challenging with viral ligases because they tend to be smaller and they tend to have a lot of non-structured domains. Okay, so without any ligands to viral E3 ligases, and also knowing that we were going to screen a variety of targets, we wanted to come up with a useful system to kind of try to assess the viral ligase capacity for neosubstrate degradation. So here we kind of we knew we wanted to use so many of the targets that we might go after won't have ligands as well. So we knew we wanted a dual tag approach where both tags are ligandable. So we kind of combined we kind of combined two different degradation systems from the literature, the DTAG system, which everyone is very familiar with FKBP12, F36V, and we put that on the target side. And then we used a lesser well-known Degron tag called MTH1. This was part of the Achilles tag system or A tag system that C4 Therapeutics published. And so this MTH1 protein, we chose it for a few reasons. It was thought to be important oncogene, and so there, historically there was a good amount of effort to come up with high affinity ligands for it. But once CRISPR was kind of brought online, it was then realized that this protein was actually non-essential for healthy cells or even most cancer cells. It doesn't matter either. So we basically have this small 18 kilodalton benign protein with high affinity ligands. And so, we fused that protein to the viral ligases, and then we made a model VIPER-TAC or a model PROTAC called FM4, which has both the ligands to those tags linked with 4-PEG. And we can co-express these two chimeras into cells and then treat with the FM4 molecule and hopefully get this chemical induced dimerization. And then we can monitor target abundance with a high bit signal. So at first I screened two different targets, BRD4 and MST2. I chose BRD4 because I wanted to increase my odds of success basically at the beginning of this project. And that's, a very well-known, easily degradable target. And then MST2 is a cytosolic kinase that is upstream in the YAP-TAZ pathway. So it's just a totally different type of protein than BRD4. And so I'd like to highlight two different kind of viral ligase patterns of degradation here. The first is this protein called V. So this is a viral ligase that's name is actually just V. And what we can see here is that there's it's good degradation of BRD4, but it's really not capable of degrading MST2 too well. So this is actually nice kind of negative data to see because we don't want all the degradation to be dominated by the tags. So this gave us like kind of a hope that the system can actually report on the compatibility of the viral ligase with targets that it's capable of degrading. It's also nice to see the type of pattern that we see with the protein E6, where both targets are equally well degraded. So we kind of had some early hopes that protein E6 might be like the viral cereblon. So I just want to show, a heat map of a summary of the screen that I performed with all of the viral ligases. This is representing the maximum degradation that was observed with titrations. And what was immediately clear is that many of the viruses are capable of degrading BRD4 to at least about 40 or 50%. So this, is good. That tells us that evolution has done its job well. And we really had something, going here. And then for the MST2 protein, it was nice to see that about, 7 of the viruses were capable of degrading from that protein as well. So, so we had this initial data set and we had to decide which one to move forward with. I chose E6 as a lead candidate due to its degradation activity seen here, as well as its medical relevance, which I'm going to get into now. So human papillomavirus is one of 7 oncogenic human viruses. This figure on the left kind of shows the viral oncogenes from these viruses and how they affect the hallmarks of cancer. And then if we cross-reference those list of viral oncogenes with the list that I made with E3 ligase activity, we can see that there's potentially 5 oncogenic viral ligases. And E6 is one of them, and I think it works in a really kind of fascinating way. So I'm showing a cartoon schematic here, but the protein E6 works as more of a biological molecular glue rather than a PROTAC. And by that I mean that the E6 protein and the E6AP human ligase, neither of them have affinity for p53 on their own. It's only after E6 binds to E6AP that there's a new protein surface that's made that recruits p53 for degradation. And so this helps bioreplication, but this is like a classic example of how these can be oncogenic as well if you're constantly degrading p53. So HPV positive cancers represent a pretty large unmet medical need, and what I believe is a good fit for a degrader modality. This is because the E6 gene actually incorporates into the human genome and drives oncogenesis, even if there's not productive viral infection anymore. And the E6 and E6AP proteins, they have a picomolar affinity for each other and quite a large protein-protein interface, which precludes small molecule inhibitors. There's been quite a bit of work to find inhibitors of that interaction, because then you would liberate p53 and the cells could go through apoptosis. But those kind of molecules that have been discovered don't really translate into even cellular assays, because this complex is preformed and very hard to break apart. So we're proposing to just let it be a complex and repurpose it. There's a huge number of cases per year globally. There's 500,000 HPV positive cancer cases per year. That's primarily cervical cancer. And in the US, there's 37,000 new cases per year, still a pretty large number. This is despite effective, safe and effective HPV vaccines being available for about 15 years. But the vaccination rates aren't what they should be. And so this is going to continue to be a problem for the foreseeable future. In the US, there's a lot of cervical cancer, but there's also a lot of oropharyngeal cancer as well. There's no targeted therapy currently available. And however, there are ongoing clinical trials that are targeted therapies, but all the modalities are biologic modalities, so either vaccines or even like viral therapies. So what we're proposing here would ultimately be a small molecule targeted modality, and it would offer all the kind of traditional benefits of small molecule therapeutics. Okay, so I hope I've convinced you that this is a valid lead viral ligase from a medical relevance perspective. And then if you think back, we chose it because it also has good TPD activity, but we wanted to make sure that TPD activity was on mechanism. I mentioned that the MTH1 tag is small, but the E6 protein is in fact smaller. And so I was kind of concerned that I might be losing the endogenous function of E6 when it's in its chimeric form. So what I did was just a simple co-immunoprecipitation, and luckily I saw that the MTH1 E6 chimera was capable of pulling down endogenous E6AP, but two different E6 mutants in this pink region of the protein here, which should prevent E6AP binding. Those still, prevented binding, which was great to see. And then even more importantly, those binding mutants have significantly reduced activity in the degradation assay. So this is telling me that MST2 degradation is dependent on the interaction of E6 with E6AP. And just to confirm this, I knocked down E6AP with siRNA before performing the assay in the cells, and you can see a nice significant reduction in the degradation activity as well. So, to summarize here, E6 degradation requires its interaction with E6AP, and then, this is still using our model substrate MST2, and we really promised to be able to degrade pan-essentials. So that was the next step, is to address that question. Okay, so because I had ventured into the oncology realm with this particular viral ligase, I decided to analyze the depth map for the to find nominate pan-essential genes. So I took the uh, average Kronos dependency score across all the cell lines in the depth map and just rank them to find this kind of subset, or what I call like the most pan-essential genes. And then I kind of manually picked out 8 of them to try to vary their biological functions. And I cloned these into high-bit FKBP12 chimeras, and included them in our chemical induced dimerization assay with E6. The translation factor SARS1 emerged as a leading candidate. This is a serine aminoacyl synthetase 1. So this is the protein that charges serine and selenocysteine tRNAs with their amino acids. And yeah, so this kind of emerged as the leading ligase target pair. So, decided it was time to engineer a more medically relevant model system. And for this, I chose the C33A cervical cancer cells. This is it's actually a HPV negative cervical cancer cell line. And I chose that because I knew I was going to need to utilize the MTH1E6 chimera. And the cell lines that like HeLa and Casci, which have E6 in them, the incorporation into the genome is at multiple loci, so knocking in would be challenging. So instead, I took this cell line and I first homozygously knocked in FKBP12 high bit to the SARS1 endogenous locus. You have to have a homozygous knock-in in order to kill the cells, but this is challenging because the FKBP12 it can't disrupt the function, or else you won't be able to have a homozygous knock-in. So the N terminus didn't work, but luckily the C terminus did to accept that tag. And after I had that cell line that was reliant on my chimera, I inserted the E6 MTH1 at the AAVS1 safe harbor locus. And to ensure I had a biologically relevant amount of E6 being produced and that it was still functional, kind of a nice and elegant way to test that was just by blotting for P53, because as I showed you earlier, this protein, this complex, which is here, is going to degrade P53. And what you can see with the constitutive promoter on my construct, but not with the parental cell line or not with a different viral ligase. Here I have almost complete degradation of P53, which told me I have a biologically relevant amount of functional protein. So then I treated with the FM4 VIPER-TAC, and by 6 hours, I was getting nice degradation, about 75% reduction in the high bit signal from SARS1. And I showed that this was dependent on the proteasome, and you could also reduce the effect by competitive tag ligands as well. In this system, I also showed that the degradation was dependent on E6AP by knocking down endogenous E6AP. And so, I was feeling pretty good about the system at this point. But again, the promise is to selectively kill cells here. And so, for the first thing I kind of looked at was, before I expressed E6 in the system, I took the parental cell line, which still has the FKBP12 SARS protein. And you can see that VIPER-TAC treatment does absolutely nothing to those cells. So if anything, it helps them grow a little faster. But when the cells are expressing the viral ligase, we've got potent, rapid, and robust cell killing here. So this was like really exciting. This was not my first attempt at engineering a system like this, and so to see this result was really the moment when I knew I proved the concept of VIPER-TACs, and And yeah, it's great. So I just want to note that the potency is rather high, and this may be because you have high affinity ligands to the tags, but it also might be because when you are degrading a translation factor, you reduce the resynthesis rate, and so you might have a positive feedback loop. I also want to note the time. The time is pretty quick for an oncology assay here, and I just want to note that even by 24 hours, there's a significant effect. So to conclude, many viral E3 ligases demonstrate some capacity for targeted protein degradation. The papillomavirus protein E6 that can degrade the pan-essential protein SARS1 on mechanism, and VIPER-TAC treatment selectively kills E6 expressing cancer cells in the chemical biology model system. So, in the future directions, definitely finding ligands for viral ligases on the to-do list. And I think it's also important to demonstrate similar proof of concepts in relevant models for other indications, other oncology indications like hepatocellular carcinoma, but also non-oncology indications as well, like Castleman's disease. And then just testing VIPER-TACs as antivirals, I think, is also going to be really important. Okay, so that I'd like to just acknowledge all my colleagues in the induced proximity platform at Amgen. And with that, I'm happy to take any questions.